All right, thank you for participating in the NIH Metabolomics Interest Group webinar series. Today's webinar from Dr. Andrew Patterson will focus on decoding host microbiota communication through metabolomics. Before introducing our speaker, I'll quickly review a few logistics. Should you experience technical difficulties, you can contact us through the questions box in the expanded control panel, or by phone or email. If you need to view live closed captioning, please click on the link that will appear in the chat box. At the end of Dr. Patterson's presentation, we will allow time for questions. If at any time during the presentation you do have a question, please type it into the question box and I will ask it on your behalf during the Q&A session. Now it is my pleasure to introduce Dr. Andrew Patterson. Dr. Patterson is an Associate Professor of Molecular Toxicology at the Pennsylvania State University and the Scientific Director of Metabolomics. Dr. Patterson graduated from the Joint National Institutes of Health and George Washington University Graduate Partnerships Program in 2006 and completed a postdoctoral fellowship as a Pharmacology Research Associate in the Laboratory of Metabolism under Dr. Frank Gonzalez at the National Cancer Institute in 2011. He then joined the Center for Molecular Toxicology and Carcinogenesis at Pennsylvania State University in 2011. He and his students, postdocs, and collaborators focus on understanding the host metabolite microbiota communication network, specifically how the manipulation of gut microbiota by diet and or xenobiotics impacts host metabolites, their metabolism, and how these co-metabolites interact with host nuclear or soluble receptors. Today he will be discussing decoding host microbiota communications through metabolomics. Welcome, Dr. Patterson.
Hi, everybody. Excuse me. Hi, everyone. Hopefully, I apologize for a little bit of a cold and coughing periodically throughout the talk today. So, okay. So, thanks everyone for joining today. Feel free to send me email at the address below, or you can always directly contact me through Twitter as well. I will talk a little bit about some drugs that we've developed through some of this investigation, so my disclosures are here. And just to lead out into this talk today, I just wanted to go back to some literature from the late 1960s. We can even reach back even further, where some of the understanding with respect to how the microbes impacted health and disease had been clearly understood. Unfortunately, we didn't quite understand how to measure them properly. And I really like this quote regarding to understanding or the importance of how techniques have really been important for understanding what the gut microbes are doing, not only in terms of how they're changing in terms of like census, population level, but also how they are impacting metabolism. So either contributing metabolites themselves or altering metabolites that may be produced by the host. And so kind of the central premise of our lab's work is summarized in the following diagram, and that is trying to understand how we can manipulate the intestinal microbiota with a variety of different approaches. So whether it's through the diet or pharmaceuticals, the easiest example might be with antibiotics. Of course, we know how this impacts the intestinal microbiota, but furthermore, also how things like toxins or toxicants that we are exposed to through our diet or through the environment can also impact this intestinal population quite significantly. Importantly, what I'll talk about today is really how we can use the metabolites that are either produced or modified by the microbiota to understand how they may be signaling to the host. And we focus just on one very specific pathway, and that is through nuclear receptors. And so nuclear receptors in this case are transcription factors that mediate a variety of effects, and I'll go into more detail about this later on. It's also just important to point out that while I end up talking mostly about that occurs in the gut, a lot of what I do talk about could readily be applied to other important niches like the skin or the lung as well. So getting back to that first quote about the need or the lack of tools that were important for studying the microbiome, really fortunately now we're at a stage where we can do this actually quite well. And so some of the advantages, have really, or some of the applications have really been, pushed forward through tremendous advances in things like sequencing. So, 16S ribosomal RNA amplicon sequencing, as well as shotgun metagenomics and metatranscriptomics, taking advantage of very unique mouse models, whether they're conventional, those with bacteria, or notobiotic, or germ-free, those with either defined or lacking bacteria altogether. But where I think metabolomics has really helped understand or help us understand this, the contribution of the microbiome has really been important for understanding the metabolism of the microbes themselves. And so in my lab, we use a variety of different platforms, whether it's NMR or LCMS or GCMS. And throughout my talk, I do try to indicate which platforms we have used, just to give you an idea for what we think is, a good tool for studying the various biofluids or extracts that I will discuss. And it's important to point out, of course, that being able to collect all this data is one thing, but really being able to analyze it in a meaningful and integrated way is another. And we really stand, kind of on the shoulder of a lot of giants to use these really great bioinformatics tools that have allowed us to more comprehensively understand the chemical repertoire of the microbial population. So again, what it's producing or what it's metabolizing, but more importantly, how we're able to take this information and really put it into a more integrated or comprehensive view of the microbiome. So beyond just understanding which microbes are increasing or decreasing in population, but more importantly, how their metabolic activity changes, and ultimately how we can use this information to understand how they're interacting with the host. And so we focus on small molecules or these chemicals that traverse from the host and the gut microbes and vice versa. But of course, this is just a small snapshot of the interaction that's occurring. But of course, for this talk today, this is one of the most critical aspects to follow. So if we carry along this line of small molecule chatter, and basically this is the way that we think we can follow how the microbes and the host are interacting. The question is, of course, how can we tap their line, or how can we listen in on the signal? And of course, this is where metabolomics comes in. And, just thinking of this in the simplest context of chemical fingerprinting. And going back to some of our original work back when I was a postdoc in Frank Gonzalez's lab, we often thought of this, these data that we were collecting as being contaminated with metabolites that we thought to be of bacterial origin. And so often we didn't really know how to interpret these results other than to know that indeed the microbes were somehow influencing the data that we were collecting. And so going back through some of our old data, we could see compounds that were indole-containing compounds or even derivatives of tryptophan, for example. And it's a really good example of a process that's mediated very carefully by the microbes in terms of understanding like serotonin metabolism, but also for immune function. Hippurate has a tremendously long history as one that is well studied in terms of its ability to track well with changes in the gut microbiome population. Cresols, glycine conjugated compounds we've followed, as well as bile acids, which I will touch on. And you can think of these, or begin to think of these as co-metabolites. And this is something that Jeremy Nicholson and Elaine Holmes put forward, this idea that these metabolites are acted upon not only by the host, but also by the microbes themselves. And so the big question is, how do we begin to define what that chemical unknown, you can also think of this, I've heard it also referred to as the chemical dark matter. But you can appreciate that, there's a tremendous amount of information that may be contained in the gut microbial population itself. Here I'm just showing a network map of a stool sample where we just profiled an extract, in this case an aqueous extract, looking for metabolites that we could identify, and those are shown in blue, and then those that we could not. And so this has all been facilitated through the work or the programs developed by Peter Dorstein out at UCSD. And this is this idea of molecular networking. And so we are able to start to get a good sense of some of the metabolites that are there, and some that, of course, that we would expect, like bile acids or breakdown products of bilirubin. We can also start to capture some things of like drugs that people have ingested, or in fact, even in some environmental chemicals that we know have been people have been exposed to. And so the development, a lot of these databases have really helped to begin to annotate some of this. But of course, we all know that these are, still being developed, but of course have a tremendously important role in helping us to define what it is we're seeing in our samples. And the reason I do end up showing this is because I do think it's important, because it does allow us to, A, try to understand perhaps what the metabolites are that the bacteria are producing, those that it's metabolizing, but furthermore, perhaps to identify targets that we could use to manipulate the microbiome itself. So could we develop small molecule inhibitors or activators that could then help to carry out some beneficial or therapeutic effect. And I'll share some of that data at the end of the talk. And so I like using this example. This is a project that we've been working on for a number of years now, but it has to do with this drug Tempol. And so Tempol is a very unique compound. It's a stabilized free radical, and has been used for a variety of different treatments. First, to help prevent or reduce the impacts of ionizing radiation. But furthermore, it's also been shown to have some preventative effects or chemopreventive effects for things like liver cancer, at least in rodent models. One of the really interesting things that had been observed with Tempol early on is that it causes really tremendous weight loss in mice. And this was an observation that was made by Jim Mitchell at NCI. And just as I was starting my postdoc in Frank's lab, he came to us with this data, which he could not explain by looking at traditional things like gene expression in the liver or changes in mouse physiology. There was no changes in food intake and things that would begin to give him some clue as to what was going on with these animals. But interestingly enough, those animals that were on Tempol throughout their life did not gain nearly as much weight as those that were on controlled diet. And this was something that had really puzzled him but intrigued him for quite some time. And he approached us thinking that perhaps metabolomics could help solve some of this problem. So at the time we had been working with this notion that we could use metabolomics in the sense of really trying to understand drug metabolism, using or taking advantage of some of the beautiful things that mass spectrometry can do, and that is by comparing Tempol, shown on the left, or comparing Tempol that in this case had been where we had modified it to include 17 deuteriums. And so in this case, what we did is we gavaged mice, we collected urine, and then we profiled these samples by ESI QTOF-MS. In this case, this was done on a Waters Premier at the time, and then profiled it to look at the metabolites, which we can see in a simple PCA plot by looking at those metabolites that come from Tempol. So we can easily compare Tempol and those that were deuterium labeled, and it was an easy way to pick out the drug metabolites from the rest of the endogenous metabolites. And so that was really great because it allowed us to expand the metabolic map of Tempol. And you may say, oh, so what? This is not all that important. However, what it did allow us to, kind of serve as a model for using this approach for other drugs. So colleagues in the lab have also used this approach to expand the metabolic map of things like common analgesics like acetaminophen. This is work done by Chi Chen, who's now at the University of Minnesota. But what you can see is that the metabolites that we knew at the time, those just shown right here in the center of this metabolic map, were well established. But using this approach, we were able to expand this metabolic map substantially. But unfortunately, this didn't really give us a good clue as to perhaps why these mice weren't gaining weight. We did see that these things were heavily glucuronidated, but unfortunately, it did not really give us much information about their mechanism of action. Now, using this approach, we were able to distinguish drug metabolites very nicely from the endogenous metabolites. And what we began to see is that over this time course of 5 days where we provided Tempol, in this case via Gavage, we could see some really interesting things that are just summarized in these two plots. The first was that if we looked at the urine of these animals, we could see a tremendous decrease in pantothenate and isobutyrylcarnitine, suggesting that there was some alteration in energy metabolism. So these are important for fatty acid beta oxidation. And really quite interestingly, we saw that as we provided Tempol, we saw tremendous changes in these quinoline metabolites, in this case 2,8-dihydroxyquinoline, as well as its glucuronide. And through some literature searching, we began to realize that perhaps these quinolines served as a beacon to indicate that the microbiome itself was changing. Whether that was a population change or a metabolic change, we had yet to determine. But this began to give us some idea that perhaps this was the key to understanding the beneficial or the metabolic effects of Tempol exposure. And so, just having revisited this in a slightly different way, we looked again at Tempol and vehicle-treated animals, and so this is just again a simple PCA, principal component analysis plot. And here we were just looking to see what metabolites were significantly changed, or what metabolites were changed between the two groups, just really using it as a screening tool to allow us to kind of evaluate the metabolic changes that were occurring. And one thing that would routinely popped up through these analyses was that the Tempol-treated animals, shown in the black squares, had depleted levels of cresol and cresol glucuronide. So you can see that those that are shown on the right, we can see that these paracresol sulfate and paracresol glucuronide levels are increased in the vehicle-treated animals relative to the control. This was published in JCI in 2015 if you'd like to see some more information. And so if you know anything about cresols, or if you've ever been around pigs, this is predominantly what that smell is that's coming from pigs. And so I grew up in Lancaster County, which is a big agricultural community, and one of the things that was in my aunt's farm where I spent a lot of my childhood were pigs, and so this was a really good indicator of where this smell was coming from. And so if you learn nothing today, you know that pigs smell like this because of paracresol. Furthermore, one of the really foundational publications in this area was work put out by the Wyckoff and Gary Schustak's group, basically comparing the differences between germ-free animals and those that were conventional, meaning those that had bacteria. And what they observed, in addition to a lot of what I already mentioned, was a variety of metabolites that they began to ascribe to gut microbial processes, and in fact were able to prove some of this by doing in vitro-based cultures of these bacteria and looking to see which metabolites were produced. And so again, I think this is perhaps one of the really important papers in the field, beginning to help us understand that perhaps metabolomics could be really important for understanding the gut microbiome or the microbiome in general. And so just to kind of convince you furthermore that these observations are indeed related to microbial activity, in this case we've just provided mice with a cocktail of antibiotics. And much like what we saw with the Tempol-treated animals, again, we could see that antibiotic-treated animals relative to vehicle had reduced levels of things like paracresol sulfate, paracresol glucuronide, as well as those dihydroxyquinolines that I talked about previously. And so this really helped to solidify this idea that what we were seeing in our metabolomics data was indicative of changes in the gut microbiome itself. And so when I got to Penn State, one of the first things we did was to profile these samples based on 16S ribosomal RNA gene amplicon sequencing. In this case, this data is based on 4x4 sequencing, and of course this is now advanced to Illumina. But one of the things that we were able to very readily see was a tremendous impact on the Lactobacillus population in Tempol-treated animals. And so this was kind of a bit of a surprise, given the fact that a variety of toxicity studies have been done with Tempol, and for the most part, they thought this compound to be rather innocuous, innocuous in particular to the host. But of course, this had not yet been considered for its ability to impact the gut microbiome itself. And so this is one of the first observations we made, and then we've gone on to follow this quite substantially to kind of understand not only how the compositional change of the microbiome occurs following Tempol treatment, but what is actually the functional consequence of that. In other words, how does the depletion of some of these bacterial populations influence metabolism? And I'll focus on that for the rest of the talk today. So one thing, just to convince you that indeed we saw changes in Lactobacillus, remind you that there are a variety of different Lactobacillus species. Here we just look at a simple culture-based approach where we isolate Lactobacillus from the cecum, the pouch of bacteria contained in mice, culture them out in pH 4 MRS broth, and then we can actually quantitate to see that indeed the sequencing results mirrored what we could obtain from mice that were treated with increasing doses of Tempol. So we could see a dose-response effect, and importantly, this was helped to really validate some of the sequencing data that we observed. So we began to wonder how was this compound Tempol impacting the microbes. We had done a tremendous amount of work to look at the toxicity of this compound with respect to the host, but unfortunately we had yet to really evaluate the possibility that these compounds, like Tempol, could also impact the gut microbiome itself. And so this is a technique based on things that I'm sure we're all familiar with, flow cytometry, that allows us to really capture some interesting things about the microbial population in terms of its overall physiology, if you will. And these are techniques that actually have been developed quite substantially from those that are studying marine microbes, and it's something that we've adopted actually through interactions with Peter Turnbull's group to help to evaluate the possibility that these compounds like Tempol are impacting the bacteria in perhaps an adverse way. And so here what we do is we isolate bacteria, in this case from the cecum of mice in an anaerobic chamber. We incubate then this bacterial population, or I should say this microbial population, with a variety of stains that allow us to look at different parameters. So Cyber Green, you're probably familiar with that in terms of its ability or its use in quantitative PCR. But we can use this as a tool to look at nucleic acid levels. We can look at CFDA, so this is a stain looking at metabolically active cells. PI, so those of you that do a lot of flow cytometry knows that this is a stain for compromised membranes. And then lastly, we can use things like dieback to look at changes in cell membrane potential and looking at depolarization of those cells as well, another indication of toxicity. So here, I want to show first the in vitro data where, again, we've taken, in this case, just wild-type animals, untreated, and isolated their fecal contents, and then incubated them with increasing amounts of Tempol. So here labeled low, medium, and high, and then also just using a positive control, in this case pH 4, to indicate that indeed these dyes are, or I should say the treatments are having a measurable effect. So if we look at changes in metabolic activity with CFDA stain, we can look at that microbial population via the flow cytometry technique. We see that as we increase Tempol levels, we see a decrease in metabolic activity. Furthermore, if we look at levels of dieback or PI, we can see an increase or changes in membrane depolarization as well as increased membrane damage. Really kind of surprising us to think that these compounds like Tempol were having a deleterious effect on the bacteria. And just to convince you that we're not seeing something that's strictly due to the in vitro assay, we can do the same thing in mice that we actually gavage with Tempol, kill them, isolate the fecal contents, and look at the metabolic activity. In this case, much like what we saw previously, CFDA or metabolic activity goes down, increased membrane damage goes up with PI and Tempol treatment. So this began to give us this, helped to solidify this notion that in addition to what we had seen in the sequencing-based approaches, that indeed Tempol was having, in fact, a direct effect on these microbial populations. And this is something, again, that has been established before with other drugs. I mean, antibiotics being a really great example, but others have demonstrated quite, nicely, including groups like Peter Turnbull, has shown that indeed these compounds can adversely impact the microbes themselves. And so this kind of gives us a new way of thinking about how things may be toxic. So whereas they may not be toxic to the host, they may in fact be toxic to the microbes. And this is an important area of concern for the field of toxicology. And so, thinking about this change in physiology, we began to wonder, could we see this change in a metabolic output? And here, as what I've shown previously with mass spectrometry, this is just an NMR-based analysis where we looked at the fecal contents, either those from the in vitro assay or those from the in vivo assay, where we can analyze these, in this case, by NMR, and then use a way of visualizing this data that has been very popular or popularized by the Jeremy Nicholson group, that allows us to kind of get a sense for how metabolites are changing or different between the two groups. And so the one thing I just want to point out is that if we look over here in this region of spectrum, we can see that relative to the Tempol-treated animals, we see increased levels of short-chain fatty acids like acetate and butyrate and propionate. However, what we can see in the Tempol-treated animals are enriched levels of oligosaccharides. And so this can start to give us this idea that perhaps this is an alteration in bacterial fermentation. So knowing that these oligosaccharides are fermented to generate things like short-chain fatty acids. This was an observation that we had made previously, but had been confirmed whether we looked at it in vivo or if we took those same fecal contents from mice, those that we had exposed to Tempol, we could see that indeed the same changes we could see very similar changes between the two groups. And so the nice thing was this allowed us to start to get or establish a metabolic signature of the activity, in this case, in the microbes themselves. And so just on the left, I just show a simple PCA plot of the changes in those populations treated with increasing amounts of Tempol. But what I want to show, or what I want to emphasize, is those that we think are really important indicators of metabolic status. And can we actually use this perhaps, or maybe eventually, as biomarkers of metabolic status in the microbiome. So, of course, this may be challenging to differentiate the microbial metabolites from those that are of host origin, but at least in this assay, it's a start. It helps to establish those that may be associated with an inactive population. So we know that these generally are inactive due to changes or increases in PI staining, or those that have changes in metabolic activity or membrane potential, from those that maybe are more associated with an active population. So meaning those that are more metabolically active and less damaged. And so to us, this is a really, kind of a good first step in trying to understand how we can use this combination of this flow cytometry technique with this metabolomic technique to get a sense of how these compounds are impacting the microbiome, whether that is a beneficial or a detrimental way. And so, for the most part, we use combinations of NMR and mass spectrometry to profile these samples. So a lot of this work had really started to center on this idea that these compounds, like Tempol, were impacting short-chain fatty acid levels. More importantly, we're impacting fermentation of these oligosaccharides to generate short-chain fatty acids. And so a lot of what we know to be, or that we know short-chain fatty acids are important for, we know that they can be an important additional addition to the caloric intake of humans. Although it is a minor component, although it can be a significant amount over time. Furthermore, they can serve as energy sources for colonocytes, or themselves can serve as signaling molecules and help to regulate metabolic and immunological function. And so one of the things that we had really been curious about, and a lot of this type of work doesn't really end up showing in a main manuscript, but we began to kind of wonder how do different approaches, whether we look at these short-chain fatty acids in their native sense or in the derivatized form. And so here I'm just showing a GCMS analysis of short-chain fatty acids that have been underivatized at the top, or those that have been derivatized at the bottom. And the reason why we began to wonder, perhaps these different approaches may impact the ultimate output of the data. And this was something that we had been quite concerned about, given that labs have different preferred approaches as well as different platforms. So perhaps this may ultimately influence the data. And so while I show here examples of short-chain fatty acids, we've now gone on to do the same things for lipid species like ceramides, as well as bile acids, which I'll talk about in a little bit. The nice thing, of course, with short-chain fatty acids, and in particular when we talk about GCMS, is that they're volatile, so we don't have to do a lot of derivatization. However, there are advantages to doing derivatization, in particular helping to have them retained a little bit longer on the column. And furthermore, we also were interested to see how these different approaches were compared to something that we would consider to be a somewhat orthogonal technique, and that is, do we see differences between measurements that we made on a GCMS versus one that we may have made on an NMR, or furthermore, could we see changes that may have been done, or see changes in measurements on, say, something that's like a LCMS system. And so we've done comparisons between short-chain fatty acid measurements on GCMS and NMR, and I'll just show a summary of some of that data here. And basically what we found, of course, that derivatized short-chain fatty acids really dramatically increase the sensitivity, and so that's shown up here at the top. There's also a very commonly or a popular method where you look at GCM or short-chain fatty acids just looking in via acidified water. This is actually a technique that's quite popular in agricultural settings, particularly those individuals that are interested in looking at bovine short-chain fatty acids. But furthermore, we're also kind of interested to see how this also translated once we did quantitative measures against using NMR relative to various standards. And so we could observe very interesting things related to how consistent these results were. So whether we were looking at the percent RSD over time, also how did this influence the recovery, and really quite reassuring is that regardless of the method we actually ended up looking at, if you looked at those short-chain fatty acids, in this case, these are obtained from measured from mouse fecal pellets, we saw that by and large we didn't see a tremendous difference in the amounts of short-chain fatty acids measured in these samples. And so this was really quite reassuring. However, there are advantages and disadvantages to using one method over the other. If you're not worried about sensitivity, of course NMR is a great technique given its quantitative nature as well as its reproducible nature over time. But furthermore, the challenges with GCMS in terms of derivatization could also perhaps influence the data somewhat, although we did not see ultimately any significant change here. And ultimately, we could apply this to a real-world scenario, in this case, just looking at short-chain fatty acids, whether we measured them in conventional or germ-free. And this was just nice validation that indeed that the sensitivity that was afforded, in this case by GCMS derivatization technique, allowed us to measure really relatively low abundance short-chain fatty acids in germ-free animals, indicated here by GF, relative to those that are found in conventional animals. Okay, so kind of switching gears a little bit here from short-chain fatty acids. So I've kind of demonstrated that indeed Tempol itself can impact short-chain fatty acid production, or in other words, fermentation. But one of the things that we had really begun to be interested in with Metabolomics was this idea that we could use this as a tool to understand how exchange of dietary nutrients, drugs, as well as things that may be of microbial origin occur between the small intestine and the liver. And so, of course, this is the process of enterohepatic circulation. This impacts a variety of things. In addition to drugs and nutrients, this can also be a really important mechanism by which some drugs become extremely toxic if they are not maintained in this enterohepatic circulation loop. And the thing that's quite interesting about this pathway is that there are a variety of host receptors, in this case nuclear receptors that line both the small intestine and the liver that we think perhaps are there to help sense that gut environment, signaling back and forth between the liver. And in this case, I'm just going to focus on one particular pathway that we know to be strongly influenced through enterohepatic circulation, and that is with bile acids. And so we began to wonder if indeed Tempol was influencing bile acids or other important metabolic pools. And so in addition to looking at a variety of other metabolites, the one that we began to focus on was, in fact, bile acids. And so just a little bit of a summary about bile acids. You know, they are produced in the liver from cholesterol, secreted into the intestines or the small intestine. And importantly, you know, historically thought to be important for solubilizing and mobilizing fat-soluble vitamins and nutrients. And ultimately, these bile acids are reabsorbed from the intestine, in this case in the ileum, and taken back into circulation in the liver. Now, a lot of this process is regulated through a variety of receptors. In this case, I will focus strictly on the farnesoid X receptor. This is also known as the bile acid receptor. And like other nuclear receptors, in this case, it's a ligand-activated transcription factor, meaning it's bound by ligand to carry out its function. And actually, quite surprisingly, if you look at the amount of bile acids that are secreted and reabsorbed on a daily basis, at least in humans, estimates range from on the order from about 20 to 30 grams of bile acids. And you know, this is one thing that's actually quite interesting, is that if you begin to look at the number of bile acids that are currently being able to measure, you know, we're actually now looking at, you know, on the order of more than 100 different bile acids. This begins to kind of allow us to start to question this idea: are these really there just strictly to help solubilize and mobilize these dietary compounds? And so the way to begin to think about this is that these are strictly not just there to act as detergents, but perhaps they actually have really important signaling properties as well. And so a lot of the bile acid profiling that we do in the lab is based on this paper published a number of years ago. We've now modified slightly, and I should mention that ultimately a lot of these protocols I mentioned, some can be found on the Metabolomics workbench, although some will be eventually up once some of these papers are published. And so it actually serves as a really great resource maintained out at UCSD for those of you that are interested in looking at protocols that are important for metabolomic analysis. So using this approach, in this case by mass spectrometry, we went back and looked at animals that had been treated with Tempol, analyzing on the left fecal bile acids, and then analyzing on the right those bile acids that we found in small intestine. And one very interesting observation was made, and that is that we found a tremendous depletion of unconjugated bile acids in the Tempol-treated animals. And if we looked actually in the small intestine, we could see a tremendous enrichment in conjugated bile acids in Tempol-treated animals. And so it's important thing to point out here when we're talking about rodents, a lot of what we end up measuring are taurine conjugated bile acids. So whether it's TUDCA, TCA, or T-beta-MCA. But if we do switch and talk about human bile acids, then we start to refer or see that glycine conjugation predominates. And so this observation was really quite intriguing and began to kind of help us start to think more about the factors that were impacting bile acid metabolism, and that has a lot to do with the microbes that are contained within the small intestine of these rodents. So a lot of the bile acids that are secreted from the liver can actually get modified by microbial processes. Here I'm just showing the dehydroxylation and deconjugation pathways that can occur with different bacteria. So in this case, just looking at Lactobacillus, we can see that they are capable of deconjugating things like taurobeta-muricolic acid to generate muricolic acid. And the enzyme responsible for carrying out this function is bile salt hydrolase. And so if you actually spend some time looking at the literature, there is tremendous information about bile salt hydrolase activity, and quite surprisingly, how varied the bile salt hydrolase activity is between, within certain genuses like Lactobacillus, but also across different genuses as well, whether you look at things like Clostridia or Bacteroides. And so, having looked at this observation with Lactobacillus, we began to start to think about that observation where we saw that Tempol reduced the Lactobacillus population by 16S sequencing as well as culture-based approaches. This began to get us really interested in thinking about how this microbial activity was impacting the host. And so one way we kind of went about this was based on genome scale metabolic models, where we use information we can glean about different bacterial metabolic processes based on their genome, and then begin to start to link this to some of the metabolic data that we've collected through our metabolomic experiments. And so I'll just summarize a little bit about what we found in this paper, and that is that as we, and this is all computationally based, of course, is that as we increase that BSH activity, we could see an increase in fermentation. This is something that we could validate in our metabolomics data. And furthermore, we began to start to think about why it is that some of these metabolites or these bile acids are metabolized by the gut microbes. A lot of it is thought to be serving as a defense mechanism. Perhaps these bile acids are toxic, but also we started to think about possibilities that they could use things like taurine to provide carbon and nitrogen sources that could, be beneficial for the growth of the bacteria. And these are things that we're actually now exploring more in more detail. And so one other thing too, I think an important observation we made, but this is one that had been somewhat established, and that is the importance of bile salt hydrolase activity in allowing or giving these microbes the capability of colonizing the intestine of animals. So here what we did is we provided, in this case, germ-free animals with different strains of Lactobacillus, one that did not have bile salt hydrolase activity, and then another two that did. And what we found, of course, is that as we looked at or tried to recover Lactobacillus that we inoculated these germ-free animals with, we found that those without bile salt hydrolase activity did not have stable colonization. And so, again, just supporting the important activity of bile salt hydrolase activity for the survival of Lactobacillus and colonization of Lactobacillus in the guts of the mice. Okay, so getting back again to this idea of microbial small molecule chatter, if we have a way of measuring it, so we looked at examples of short-chain fatty acids, we looked at examples with bile acids. So the question is, who's listening? And in this case, I mean by the host. And so the one thing that we focused on were these metabolic sensors that are part of the larger nuclear receptor superfamily. And these are PPARs, LXR, FXR, PXR, and CAR. And the one thing that I had already kind of indicated is that we began to really start thinking about the role of FXR, given what we had observed with these changes in bile acids. And so the FXR has a really well-established history as obviously an important regulator of bile acid metabolism, but a lot of really interesting data has been coming out about its important role in glucose and lipid metabolism as well. And so in thinking about this idea that FXR was involved, we began to go back, we went back and looked at the gene expression of FXR as well as its target genes in two different tissue compartments. The first, shown on the left, is looking at intestinal FXR level or gene expression as well as its target gene activation, in this case looking at SHIP or FGF15 or IBAP, and then comparing that to the same genes that we could observe or measure in the liver. And what we found was that as we looked at Tempol-treated animals, we could see that they had reduced levels of FXR target gene activation in the intestine, but something that was not observed in the liver. And this began to start to help us think about the possibility that disruption of this process in terms of the microbiome by Tempol was impacting FXR activity. And through a variety of different experiments, and actually one that had been published out of Friedrich Backhead's group as well, and that is that these bile acids actually are really potent antagonists. So whether we look at the classical agonist of FXR or a classical agonist of FXR like TCA, we can look at target gene activation, in this case just looking at SHIP in cultured hepatocytes. We treat with SHIP with TCA, we can see an induction of SHIP mRNA. However, when we do the same experiment or looked at it, the induction of SHIP with TBMCA, we could see that there was a dramatic reduction in its gene expression. And quite interestingly enough, if you look at levels of TUDCA, we know that in humans, UDCA is a very common treatment for things like fatty liver disease, or is emerging as an important treatment. And perhaps this serves, or actually people have now begun to speculate that the metabolism of UDCA may impact FXR activity, much like what I'm about to show you in the next few slides. So if we went back to some of the original data looking at intestinal levels of these bile acids, we noted again that Tempol treatment increased quite dramatically the amounts of taurine-conjugated bile acids. In particular, the ones that we began to key in on were taurine beta-muracolic acid. And we found that this was directly its levels rose as lactobacillus levels decreased. And so, again, knowing what we knew about the bile salt hydrolase activity, this began to help us formulate these hypotheses that, in fact, that this lactobacillus was controlling FXR activity in perhaps a really interesting way. And so there is some controversy in the field, and that is really, kind of centers on this idea of whether FXR activation or agonism versus FXR antagonism is beneficial or not. And through most of our research, we found that FXR agonism, at least in the animal models that we've used, can increase things like inflammation, can help to promote obesity and insulin resistance, as well as to promote things like fatty liver. And I'm just going to show you just one example of that. In this case, we've given, floxed FXR wild-type mice a high-fat diet that we know to promote the development of steatosis, or the deposition of lipid in the liver. And you can see, if we look at H&E, you can see these, these voids where lipid once was, which is then ultimately confirmed by looking at oil red O staining. And then also, if we look at the FXR knockout animals, in this case looking at intestinal knockout mice, we can see that these mice are resistant to that high-fat diet-induced steatosis. And so this began to give us this, further help confirm this idea that intestinal FXR was really important for driving some of these really interesting phenotypes in the liver. And this really serves as the foundation for some of the pharmacology that I will describe in the next few slides. And so we started to kind of toy with this idea that some of these lipids that we are observing in these animals were perhaps toxic. And so this is one of the world's foremost expert in sphingolipid metabolism. That's Al Merrill, down at Georgia Tech. And what he's been able to demonstrate quite nicely, and using his approaches, we've actually been able to very nicely and quantitatively profile some of these ceramides, or these very hydrophobic, waxy lipids that we think are detrimental to the development of things like fatty liver disease, as well as NASH and further complications of the liver. And so if we look at the levels of FXR in terms of the wild-type treated animals, or the wild-type high-fat diet treated animals, or those that we provided with Tempol, we can see that the wild-type animals given vehicle had increased levels of a variety of different ceramides, whether we looked at the ileum or the serum. But if we treated it with Tempol, so again, this compound that we knew to reduce the incidence of fatty liver in these animals, we could see a decrease in ceramides. Or if we looked at the intestinal knockouts, whether we looked at the vehicle or Tempol treated animals, we could see that these mice were also resistant to that increase in ceramide levels as well. And so the really interesting thing is that, making mice or treating mice is one thing, but really trying to establish the relevance of this pathway in humans was something that we were really intrigued by. And this was done with one of our collaborators, actually a former postdoc of Frank's, who's now out at Peking University, and who's really been kind of driving a lot of these observations in human populations. And so if we looked at a population of humans that we were able to stratify based on BMI as being lean or obese, we could see that in the obese individuals, if we looked at ileum biopsies, we could see that they had increased activity of, or increased FXR target gene activation, and this positively correlated with BMI, whether we looked at ship-RNA or message or FGF19 message. And so, this is not, fully nails the mechanism, but it does start to give an indication that perhaps a lot of what we're seeing in our mouse models, we could perhaps recapitulate in, or it could be recapitulated in the humans as well. And so now we started thinking about this process, with this pathway, in the opposite sense, and that is that if FXR agonism is bad, perhaps FXR antagonism is good, and this is where we started to think about developing antagonists of FXR and allowing us to treat this disease at a distance. So knowing that a lot of the activity of FXR in the intestine was beneficially or adversely impacting the liver function, we began to think about ways of developing new compounds. And so TBMCA we had already established was an FXR antagonist, and then we began to think about other ways of looking at or developing FXR antagonists, whether we looked at, in this case we looked around 96 derivatives. I only show a few here just as an example. In this case we looked at glycine conjugated uracilic acid or alanine conjugated uracilic acid. And quite to our surprise, we were able to identify that, in fact, glycine uracilic acid was a really potent FXR antagonist relative to what we have seen with TBMCA. And so we could confirm this by looking at in vitro studies where we competed away activation, in this case with CDCA, or the FXR agonist, synthetic agonist GW4064. And so as we increased the amount of Gly-MCA, you could see that FXR activation was decreased. And so here's the problem, and this is why TBMCA would not serve as a really good therapeutic agent, and that is, kind of as I already hinted at, it can be metabolized by bacteria. And so if we incubate TBMCA with extract containing bile salt hydrolase obtained from the cecum of mice, we can see that TBMCA gets metabolized to uracilic acid, whereas glycine uracilic acid is completely resistant to this activity. In fact, we've gone on to look at a variety of different bile salt hydrolase enzymes to ensure that this is a very robust and stable compound. And the other thing that's actually quite interesting or important to this story is that the activity we were hoping to keep reserved to the ileum. There's been a lot of data from FXR knockout animals, in this case whole body animals, showing that these mice long-term are prone to the development of things like cancer. However, looking at it from a different angle where we could specifically or tissue-specifically target our drug to FXR, this was perhaps a really, a viable option for treating things like fatty liver disease. And what we found with Gly-MCA, it was retained in the small intestine, where we saw very little of it in the liver or serum of animals treated with Gly-MCA. So looking at its effect in a high-fat diet-fed mice, we could see that Gly-MCA-treated animals had reduced weight gain. We could furthermore see that they had reduced fat mass, whether we looked at fat mass alone or the ratio of fat to lean mass as well. And furthermore, we could see that overall liver function or liver the impact of the high-fat diet on liver was also markedly impacted by Gly-MCA. We could see reduced liver weight, as well as a dramatic reduction in hepatic triglycerides of animals treated with Gly-MCA as well. And so diet-induced obesity was one thing, and this is an analogy I borrow from Frank Gonzalez, and that is, if we can use it, can we really, we can use it in a diet-induced model, can we use it to stop a freight train? And by that, I mean, can we use it in a rather dramatic way, in this case, treatment of, in this case, DB mice with Gly-MCA. And much to our surprise, we could actually see that Gly-MCA resulted in a marked decrease in body mass of these DB mice. And furthermore, we could see a dramatic improvement of overall liver function. We could see reduced steatosis in the Gly-MCA-treated DB mice, and this was, confirmed by looking at hepatic triglycerides as well. Liver enzymes, furthermore, were dramatically depleted or decreased in Gly-MCA-treated animals, again supporting the notion that this was helping to improve overall liver function. And so, in kind of conclusion, in summary, I hope to have shown you a good example or several examples for how we've used metabolomics to inform us about the process by which the microbes are influencing host activation. In this case, whether I focused on short-chain fatty acids, but in this case, just showing a summary of how bile acids, excuse me, are important for modulating FXR activity. And so, as I mentioned, these get secreted from the liver into the small intestine after a meal, and can either A, be metabolized by the microbes, or if we deplete those microbial populations, we can increase the production or the concentration of these bile acids and help to impact FXR activity. But in this case, in the untreated example, without activation of or antagonism of FXR, we can see its activation leading to the overproduction of these toxic ceramides, which can help to contribute to things like obesity and insulin resistance. And in the examples I showed here, the increase in steatosis of these animals. And so just one last thing, and kind of summarizing where a lot of this work has gone and where it's going. We know that things like Tempol, as well as antibiotics, can dramatically influence the gut microbiota. This can influence bile acid pools. People have shown this not only for kind of the context I've shown it, but also for the development of hepatocellular carcinoma. So reducing the production of toxic bile acids like deoxycholic acid or lithocholic acid. And furthermore, we've developed a variety of compounds that we know can bypass the gut microbial process and influence directly intestinal FXR signaling, which we think serves as a really unique and novel way of modifying things like obesity, insulin resistance, and fatty liver disease. So just in the last minute, just to acknowledge some really important collaborators. Importantly, a lot of this collaboration, a lot of the main collaborations done with Frank Gonzalez at the National Cancer Institute, as well as his really talented postdocs, Tao and Carol, his extremely talented mass spectrometrist, Chris Krause, and of course, we wouldn't be where we were without the initial observation by Jim Mitchell and his group. Within my own group, talented postdoc Li-Min Zhang, as well as graduate students Bob Nichols and, again, another talented mass spectrometrist, Phil Smith. People down at Hershey, Chantu, and DeMont for generating or synthesizing these FXR antagonists, as well as those in chemical engineering, Costas, Josh, and Maggie, who helped us really start to understand the possibilities of using genome-scale metabolic models to understand our data better. And lastly, just to thank the funding institutes, Penn State, the Pennsylvania Department of Health, and as well as the NIH. So thanks very much for your attendance, your listening, and I will be happy to take any questions.
Okay, great. Thank you so much for a very informative presentation today. We do have a couple of questions. First off, if you do, we'll start with the questions we've already received. If you do have a question, please feel free to type it in the question box. The first question is, what is your opinion about the importance of the microbiome in the determination of a host metabolome compared to other environmental exposures, which would be an example would be smoking?
Yeah, so I mean, this is a really interesting question, and this is one that I think is something that's starting to be developed with things like the exposome, for example. This is this idea that maybe perhaps we've overlooked the potential of environmental chemicals to impact the microbiome in ways that we have yet to fully appreciate. And so some of our work, our new work now, funded through NIEHS, is to look at some of these questions, and that is, how do things like environmental chemicals signal through the gut microbiome and impact another important receptor, which I didn't talk about, in this case, the aryl hydrocarbon receptor. But I think a lot of this work through understanding the exposome and what it is, what chemicals we're exposed to, how much, and what the ultimate impact, I think, is something that is really important to study. Although I don't know if there's actually been definitive evidence to suggest that this is possible. There's been some work with arsenic as well as mercury, which we know to be modified through environmental microbes, but ultimately how that is influenced through the host microbiome, I think, is still something that has yet to be determined.
Great, thank you so much. The next question is, have any studies been done on the impact of Tempol treatment on lifespan and or health span?
Yeah, that was a study. This is something that is still, one of the main interests of Jim Mitchell at the NCI, and so this is something that he's been exploring, kind of as you could think of it almost as like it's not quite caloric restriction, but I think it's along those lines. In fact, one of the things that he spent a lot of time trying to figure out was, was the observation that Tempol-treated animals were impacting weight gain, was it just a toxic response? And I think I did mention it, but he hasn't seen something like that yet. I know that Tempol has been explored by a variety of maybe smaller biotech companies for applications such as that, although I don't know really the outcome of it.
Great, thank you. I think we might have time for one last question. So you touched on a bit how the mice models might represent what would happen in the human. What other plans do you have to further explore these findings in a human model?
Yeah, I mean that's a really important question, because how well the human, or I should say the mouse microbiome models the human, I think is something that is intensely debated. We're really good at making mice skinny. We're really good at curing mice, but how that ultimately translates to humans, I think, is a big question.